Dissemination and invasion. Let's move to the first part of this presentation, Introduction to Metastasis. What is metastasis? Metastasis can be defined as the spreading or development of a tumor, cancer cell, from the primary tumor to other parts of the body. Here, you can see the difference between normal cell development and cancer cells development. During metastasis, cancer cells spread to other organs or tissues by invading surrounding tissues. And then, it gets into the bloodstream or lymphatic system. Lastly, establishing secondary tumors along the way. Next, there are 5 major steps of metastasis. But in our presentation, we will be presenting on the first step, which is dissemination and invasion. Dissemination and invasion, known as cancer cells, acquire the capacity to invade neighboring tissues at this stage, allowing them to break out from the main tumor and enter the surrounding microenvironment. Next, let us take a look at an overview of what is dissemination. It first begins with the cancer cells detach themselves from the primary tumor, and then invade and migrate through the adjacent tissue that will form new tumors in other parts of the body. The epithelial cells has the characteristics of tightly bound to each other and to the neighboring extracellular matrix and also a motility. These made it hard for the cancer cells to invade and migrate through the extracellular matrix, which are already complex. So the epithelial cells will go through a series of morphological and biological changes to convert from epithelial phenotype to mesenchymal phenotype. This process called as epithelial-mesenchymal transition, EMT. During EMT, the cancer cells will lose their adhesion ability to the adjacent cells and then able to leave the primary tumor site, and these cells will become more invasive and motile. This transition is very crucial as the mesenchymal cells has the characteristics of loose junction, front-back polarity, motile and lastly migratory. Comparing to the epithelial cells, which has the characteristics of tight junctions, apical-basal polarity, amotile and non-migratory. So overall, here is the primary cancer tumor site that went through the epithelial-mesenchymal transition that finally able to go through the extracellular matrix and goes into the adjacent tissue. Moving on to the third part, mechanism of invasion. The first part in mechanism of invasion is expression of cadherins and catenin in epithelial-mesenchymal transition. It is divided into 3 parts such as E cadherin, N cadherin, and last beta catenin. Let's see E cadherin. What is the EMT? EMT is the reversible process, in which a motile epithelial cells, tightly bound with one another, and the surrounding ECM, develop the ability to transition towards a more mesenchymal phenotype. E cadherin is one of the critical markers of cell-to-cell adhesion. The main function of E cadherins is to mediate calcium-dependent cell-cell adhesion. They play a key role in maintaining tissue integrity by forming adherens junctions between adjacent epithelial cells. In epithelial tissues, E cadherin is prominently expressed at cell-cell junctions, contributing to strong intercellular adhesion. In EMT, epithelial cells could go acquire mesenchymal characteristics following the dissociation of cell-cell junctions and suppression of E cadherin. E cadherin downregulation is a characteristic feature of EMT. This reduction weakens cell-cell adhesion, facilitating the detachment of epithelial cells from neighboring cells. Loss of E cadherin in cancer cells leads to metastatic dissemination and activation of several EMT transcription factors. Next, N cadherin. N cadherin serves as an indicator of ongoing EMT, and its expression has been correlated with the development of various types of carcinoma. N cadherin promotes angiogenesis and maintains the integrity of blood vessels by unsheathing endothelial cells and mural cells for stabilization of microvessels. N cadherin is typically expressed in mesenchymal tissues and is less prevalent in epithelial tissues. Like what I state in E cadherin, there is downregulation of E cadherin, so that will make N cadherin is upregulated during EMT in cancers, and this process is called cadherin switch, that associated with enhanced migratory and invasive traits. That will make E-cadherin-mediated adherens junctions dissociate, due to the downregulation of E-cadherin, while N-cadherin junctions establish a relatively weak adherens junction. Lastly, E-cadherin. The cadherin-catenin complexes are crucial for the ability of the cells to adhere to each other. The cadherin-catenin adhesion is the essential component of adherens junctions that maintain tissue stability and dynamic cell movements. Any dysfunction of the cadherin-catenin complex reduces cell adhesion. The function of beta-catenin is equally important, because its association with cadherin links the adjunction to filamentous actin and promotes the reorganization of the actin cytoskeleton. During EMT, cleavage of E-cadherin leads to the destabilization of adherens junctions and the release of beta-catenin to function as a transcriptional activator for cell proliferation. Now let's move on the role of extracellular protease to degrade extracellular matrix. To invade through the ECM, activation of proteolytic enzymes such as metalloproteinases, MMP, functions to degrade the ECM and its constituents. MMP is elevated in cancer cells and can affect tumor angiogenesis and proliferation by activating or deactivating several growth factors. Lastly, cell motility and chemokines. It is an important factor in metastatic invasion and dissemination, since they enable cancer cells to spread from the original tumor to nearby tissues and other areas. Cell motility refers to the capacity of cells to move. It is the ability of cells to change their position, and it plays a crucial role in various physiological processes, including development, immune response, and, in this context, cancer cell invasion. There are several factors responsible for changes in cell motility, such as modifications in the cytoskeleton, cell adhesion molecules, and signaling pathways. Moving on to the next part, which is chemokines. Chemokines are a type of signaling protein or molecule that cells use to communicate with each other. By attaching to certain receptors on the cell surface, they control cell motility, which is ability of cells to move, and chemotaxis, movement of cells in response to a chemical gradient, by triggering signaling pathways. Chemokines play a crucial role in two specific processes related to metastasis, which is intravasation and extravasation. The last part is molecular and cellular factors. During metastasis, a number of molecular and cellular variables affect invasion, including cell adhesion molecules, matrix metalloproteinases, MMPs, and epithelial-mesenchymal transition, EMT. So firstly, cell adhesion molecules. It is an important part of cancer cell invasion is the expression of adhesion molecules, such as selectins, cadherins, and integrins, changing. Cancer cells can interact with surrounding cells and the extracellular matrix by breaking out from the main tumor through altered adherence. Secondly, matrix metalloproteinases, MMPs. MMPs are a class of enzymes that facilitate the invasion of cancer cells by breaking down the extracellular matrix. It is functioning by helping tear down barriers so that surrounding tissues can be invaded by cancer cells. And lastly, epithelial-mesenchymal transition, EMT. It is a process by which cancer cells change from an epithelial to a more mesenchymal phenotype, acquiring improved migratory and invasive capabilities. Gene expression alterations are a part of this process. Mesenchymal markers are upregulated, and epithelial markers are downregulated. In conclusion, metastasis initiation involves dissemination and invasion as the first critical step. Molecular factors like E-cadherin, N-cadherin, and beta-catenin play key roles in cancer cell behavior during this stage. Extracellular proteases, particularly matrix metalloproteinases, MMPs, are activated to break down the extracellular matrix, facilitating invasion. The epithelial-mesenchymal transition, EMT, marks a transformation in cancer cells, enhancing their motility and invasive capabilities. Chemokines, cell motility, and various molecular components contribute to the complexity of invasion. Understanding and targeting these processes are crucial for developing effective strategies against metastasis. That's all for dissemination and invasion of metastasis. Thank you for listening, and have a nice day.